Hello, my name is Tim Stutt, and I'm the Editorial Director for Drug Discovery and Development magazine. I want to welcome everyone to today's webinar sponsored by BMG LabTech titled Enhanced Protein-Protein Interactions in Living Cells Using the NanoBRET Assay and Clariostar. This webinar will highlight the performance of the Nanoluck LuCFRET assay from Promega on the new Clariostar multi-mode microplate reader from BMG LabTech. Among its many features, the Clariostar has a revolutionary new type of LVF monochromator that has adjustable bandwidths from 8 to 100 nanometers. In this webinar, we'll learn about Promega's novel NanoBRET technology using Nanoluck and Halotag, application of NanoBRET measuring protein-protein interactions in living cells, wavelength scanning and luminescence assays, and how to enhance all of your assays with the Clariostar microplate reader. Following the presentations, we will have a Q&A segment. I encourage everyone listening to submit questions at any time during the presentation by typing your question into the designated section on your screen. We will get to as many questions as we can during the time allotted. If we don't get to your question, we will be sure to send a reply to you via email after the webinar. The presentation will be available on demand tomorrow, and you will be sent a follow-up email with a link to access the presentation anytime you wish over the next few months. Today's speakers include Dr. Thomas Microplate, who is head of the Cell Biology Advanced Technology Group from Promega Corporation. Previously, Dr. Microplate held positions in research and development at Life Technologies and SADA Therapeutics, Johnson & Johnson Pharmaceutical Research and Development. He earned his PhD in immunology from the Technical University of Munich in Germany, his postdoc at UT Southwestern Medical Center, and an MS Biology from the University of Tübingen. Also presenting today is Dr. E.J. Dell, who is the International Marketing Director for BMG LabTech GmbH and has been with the company for 7 years. Previously, he was an application scientist and a business development manager for BMG LabTech. He holds a BS in chemistry from the Pennsylvania State University and earned his PhD in biochemistry from the Northwestern University Medical School in Chicago. He has previously worked as an adjunct professor, a toxicologist, a science writer, and an organic chemist at various places including Pfizer, DuPont, and Penn State University. And now let's begin our webinar with Dr. Microplate.
Thank you, Tim, for the introduction. I would like to thank the organizers for giving me the opportunity to present my work here. I will be talking today about Promega's new emergent NanoBRET technology platform for the analysis of protein-protein interactions in living cells. So the first slide shows basically generally how BRET works. BRET is just a particular version of resonance energy transfer. And resonance energy transfer is a physical phenomenon in which energy is transferred in a non-radiative fashion between two lumiophores. Now, what makes it so suitable for measuring protein-protein interaction is the fact that this energy transfer happens only over very short distances. As a matter of fact, you won't see much energy transfer happening in distances greater than 10 nanometers. In addition, energy transfer is also exquisitely sensitive towards changes in proximity or distance. Energy transfer efficiency is inverse proportional to the 6th power of distance. Additional factors that play a role in the efficiency of energy transfer is the geometry of the dipoles of both lumiophores and then spectral overlap. Next slide, please. So in this first diagram, you see basically how the spectra of the two lumiophores align with each other. In blue, you see the donor, the emission spectrum of the donor, and of course, as opposed to fluorescence energy transfer, in bioluminescence energy transfer, you have only the emission of the donor, and you don't need to concern yourself with the excitation of the donor, since the donor is a luciferase. In the dashed green line, you see the excitation of the acceptor. In the solid green line, the emission of the acceptor. And then the cross-hatched area denotes the spectral overlap between the donor emission and the acceptor excitation. Next, please. Next. The second diagram shows it's a typical BRET diagram. You see the characteristic two peaks. The peak to the left is the donor peak. The peak to the right is the acceptor peak, which is essentially dependent or shows the actually energy transfer. And the way you measure BRET is essentially you detect or you analyze the donor signal. You can do that by using a bandpass filter that captures part of that spectrum, and then the acceptor signal, and you use either a bandpass or long pass filter to catch basically the acceptor signal. And then the acceptor signal over the donor signal is typically denoted as the BRET ratio. But you see overlaid in this diagram also in the as the blue hatched line or blue dashed line, the donor emission. And you can see, and that is the blue cross-hatched area, that there's a considerable amount of donor signal bleeding into the acceptor signal or into the acceptor channel. And that acceptor, that donor bleeds through is essentially the background. Next slide, please. And what is typically done is to correct for this background by removing the ratio, the BRET ratio of the donor from the BRET ratio of your actual sample. And that is typically called a corrected BRET ratio. Now, there are a number of advantages for the analysis of protein-protein interaction that BRET has over alternative methods, and I'm trying to outline them here. Next, please. For one, it allows the real-time measurement of protein-protein interaction in living cells, as opposed, for instance, alternative methods for protein-protein interactions such as mass spectrometry, pull-down experiments, or even time-resolved spread. Neither of these methods can measure in living samples. The second is that BRET is in general a ratiometric assay format which insulates against variations in cell number and expression, as opposed to intensity-based formats which are sensitive to all of these variations. And an example for that would be protein complementation. Next. As said before, it's extremely sensitive towards changes in distance, so you can very clearly denote the distance between two complexes. On the other side, mass spec and pull-down experiments are likely to pull down everything that is even loosely attached, so there is a certain amount of noise you're pulling through. Next. Another advantage is that there are no physical interaction between donor and acceptor, so there is no influence of your measuring system on the kinetic and the affinity of your two partners. That is, for instance, can be a problem in protein complementation assay. Next. As in contrast to fluorescence energy transfer, the donor bleed-through is the only source of intrinsic background, and that intrinsic background can be expressed as a fixed ratio, so essentially you can turn it into a coefficient that you subtract from your values. In contrast, BRET struggles or has a problem with the inadvertent excitation of your acceptor by the donor excitation. This is something that is far more difficult to control against. Next slide, please. Now, no method is without challenges and limitations. And next, please. One of the main challenges in the current application of BRET is the relatively poor signal strength of current donors, mostly ALAC and ALAC8, so basically derivatives of vanilla luciferase are used. And that comes, and that poses some problem and comes with a number of consequences. Next. For once, if you have a poor donor signal, you generally need to maximize spectral overlap, because in general, your acceptor signal will be far weaker than your donor signal. So if you have a relatively poor donor signal, you need to preserve or need to transfer as much energy as possible in order to stay in the detectable range for your acceptor. That, on the other side, will result in a limited dynamic range, which I will further explain in a few slides. It also requires relatively high donor expression levels. Once again, if you have a relatively weak signal, you can boost that by expressing your reporters at high enough levels. However, that comes with a number of liabilities. For one, high expression levels are often associated with biological artifacts. And that also provides excessive free donor, which contributes to background. Next. Once again, if you need high expression levels, you're pretty much limited to easy-to-transfect cell lines, or you have to go through the pain to essentially make stable cell lines. Next. A more principal limitation of using BRET is that you require engineered reporters and engineered cells. So there is currently no way that you can read out endogenous proteins. That is, of course, in strength of pull-down experiments and mass spectrometry. So if your goal is to read out interactions of endogenously expressed proteins, BRET isn't a method that's going to work for you. Next, please. Now we'll give you a brief overview over the constituents of our new NanoBRED technology. So on the donor side, we have the new Nanoluck Luciferase, which was released by Promega about a year ago. It is a very bright luciferase with glow kinetics. It is about 100 times brighter than ARLAC and about 30 times brighter than ARLAC8. It's a very small reporter, which is, of course, desirable, so there is less chance of the reporter to sterically impair interactions or impair functions of proteins. It has extremely high physical stability, and that I will show you a little later how that is advantageous. And it's highly adaptable for high-throughput formats. Next. On the acceptor side, we use the HaloTech labeling system. That gives us, of course, the opportunity to optimize our acceptor because the HaloTech labeling is a protein fusion tech that is engineered to covalently bind in synthetic HaloTech labeling and chloralkane derivative. The binding is very rapid and very selective. And we selectively screened a large number of different ligands, and we came up with a ligand we call now NanoBRED 618, which is spectrally well resolved from the donor, Nanoluck donor. And you can see that in the diagram on the right-hand side. You see the donor emission in the black curve, and then the acceptor excitation in the dashed green curve, and the acceptor emission in the red curve. And you can see that the spectral overlap is relatively small, but keep in mind that you have so much more light available with Nanoluck that you can afford to sacrifice some of the light in order to achieve better spectral separation. Next slide, please. Next. Now I want to talk about some of the advantages of NanoBRED technology compared to existent BRED technology. For once, it is, you have a large spectral separation between acceptor and donor, which results in an improved dynamic range. Next. The bright donor signal, of course, enables you now the use of relatively weak promoters or diluted DNAs for physiological expression level. It allows you the use of small cell numbers if you have precious samples, or alternatively high-density plate formats like 384 well plates. Or it allows you to use difficult-to-transfect cells, for instance, like hematopoietic cells. Next, please. Also, the HaloTech gives you an intrinsic negative control. You don't need to do a separate donor transfection, which you have to do if you use, for instance, the vanilla YFP system, because YFP is, of course, always on. So you have, in order to determine the donor background, the donor alone background, you have to conduct a separate transfection with the donor alone. Now, in the case of using the Nanoluck HaloTech system, you don't have to do that, because the only thing you have to do is you have to plate a few extra cells and you omit the HaloTech ligand. So you have effectively a no acceptor situation there. Next slide, please. Now I want to talk for a moment about the advantages of increased spectral separation. So here you see an example, and these were conducted with a direct fusion between Nanoluck and HaloTech. And in this first diagram, we labeled the halo tag with Oregon green, which is, of course, a fluorescent derivative. It excites around 490, emits around 520. So it's very similar to the situation you would encounter with using, for instance, vanilla and YFP. And you see basically the BRET peak or the BRET spectrum in red. You see the typical two peaks. Underlying in the blue dashed line is the Nanoluck, the donor alone. And then we assumed simply that we would have a long pass filter at 520. So basically we're measuring our acceptor signal everything above 520. And you see you get a bright, strong signal in the acceptor channel, and you can see the green cross-hatched area. That would be basically the measured signal. But then in the black cross-hatched area, you see there is a substantial contribution of your background. Now keep in mind that this is a somewhat artificial system in which we directly fuse Nanoluck and halo tag, so it won't get much better than that. In most cases, you will have a much weaker acceptor signal to deal with. However, the donor signal will stay the same. In contrast, next please, if we do the same experiments with our NanoBRET 618 ligand. Now, as expected, you will get a much smaller acceptor signal, as evidenced by the smaller peak on the right-hand side. However, you see that the donor bleed-through is barely visible. So relatively speaking, the amount of background relative to the acceptor signal is much weaker. Now, we can express that as a signal-to-background ratio. Next, please. And you see that the signal-to-background ratio for Oregon green is way lower than for NanoBRET. As a matter of fact, it's about 10-fold lower. Next slide, please. Now, I would like to proceed to show you some application examples and some comparisons to BRET1. So in the beginning, for the first set of experiments, we used a tried-and-true system, which is basically FKBP and FRB. FKBP is on cyclophilin. FRB is the FK506 binding domain on mTOR. Now, in the presence of rapamycin, FKBP and FRB form a very tight ternary complex and is very commonly used to test protein-protein interaction technologies that way. Next. In this experiment, you see basically the difference between using the NanoBRET system and BRET1, which is RLUC8, the RLUC8 YFP combination. In red, you see dose response of rapamycin applied to cells that express FRB, NLUC, and FKBP halo tag, which have been labeled with the NanoBRED ligand. And you see a strong signal-to-background ratio reaching about 10. In contrast, if you use the BRET1 system using FRB RLUC8 and FKBP YFP, you see a curve, and if you would blow it up, you see it's a usable curve, but you see that the signal-to-background ratio is considerably compressed. Next. Next. Now, one of the things that one has to do if you want to design an assay for protein-protein interaction, you have to go through an empirical optimization process of the best possible BRET pair. And I show you here on the left-hand side basically all the combinations of acceptor and donor fusions that you can that you have to test in order to find the best possible combination. Next. And you see here for both BRET1 on top and NanoBRET on the bottom that there is really one combination that sticks particularly out. But you see that also that with NanoBRET you will find a lot of combinations which still shows you some weak BRET signal. Now, ideally you would always take the strongest combination, but sometimes that is not possible for a number of reasons, and you might be forced to use a version that is suboptimal. And you can clearly see with the NanoBRET, you will see with most BRET pairs a detectable signal that is above background, while a lot of the BRET1 pairs, the YFP RLUC8 pairs, show basically disappear in the background. And that is what we believe where spectral separation really makes a difference. Next, please. Now, I talked before about the ability to use NanoBRED at low expression levels. And we tested that hypothesis by basically diluting the DNA out. And we have on the left-hand side the NanoBRED system with Halotech and Nanoluck. And on the right-hand side, the BRED1 system with YFP and ALAC8. We use the same FKBP and FRB pair. And you see basically in titration of DNA, the signal becomes weaker simply due to the fact that you start forming more sort of non-BRED pairs with endogenous proteins. But you see clearly that you get to far lower expression levels with the NanoBRED system compared to the BRED1 system. Next. And that can be evidenced by basically using a method that is commonly used in high throughput screening by determining the Z-prime score. The Z-prime score gives you roughly an estimate of robustness or reliability of detection between two different samples or to a sample and a negative control. And typically considered a Z-prime score of above 0.4 is considered acceptable for a robust assay. And you can clearly see that the Z-prime scores for NanoBRED are superior compared to BRED1, meaning that you can use far less DNA and far lower expression levels compared to the BRED1 system. Next slide, please. Another aspect is that the use of cell lines that are not easily transfectable. Of course, a lot of work is still done in cell lines that are easy to transfectable, like CHO cells, HEC293, and HeLa cells. But increasingly, researchers are interested to using cell lines that are more reflective of the actual physiology of the topic they're interested in. Now, one example, for instance, are hematopoietic cell lines. In this case, we use the Dürker T-cell line or T-cell lymphoma line, which is notoriously difficult to transfect, like most hematopoietic cells. And here we compare basically the FRB FKBP system either in its NanoBRED configuration, and you see a nice rapamycin dose response curve. And next, compare that to the BRED1 version, the FRB ALAC8 FKBP YFP combination, which essentially gives you only noise. I think, and that underlines, I think, the power of NanoBRED for the use in situations that are suboptimal. Next, please. Now, another aspect is that, of course, a lot of protein interactions are transient, and there would be certainly an advantage if one could fix that in time, especially if you want to do a conductance screen, and you need to basically align the processing of the plate with your individual process steps. So we had an idea, because Nanoluck is such a stable enzyme, to see if Nanoluck would actually survive fixation procedure, and it turns out it does. As a matter of fact, the fixed sample here in showing the green dose rapamycin dose response seems to be qualitative almost better than the non-fixed sample. That's not in general, this is just a sample experiment, so I wouldn't want to give the impression that it's always the case. But on the other side, next, yes, if you see the ALAC8 YFP sample, you see that basically the the fixed sample doesn't give you any dose response anymore. And that's simply due to the fact that if you treat Nanoluck with PFA, with paraformaldehyde, if you fix it, you take a loss of about 80% of the signal, but the enzyme is so bright it is still far brighter than ALAC8. On the other side, if you treat ALAC8 with paraformaldehyde, you lose about 99.5% of your signal, which effectively kills its activity. So it is not usable in this context. Next. Now, another interesting aspect that is tied to the brightness of Nanoluck is that it allows you for the first time to do real-time image-based protein-protein interaction analysis in single cell. And we go back to the tried and true FRB, NLUC, FKBP, Halotech system. And in the first panel, you see untreated cells. They have not received any rapamycin. On the left-hand side, you see the donor channel, which is essentially a bandpass filter, and the right-hand side, the acceptor channel. Now, once you add rapamycin, you see clearly the acceptor signal coming up. Next. And this was part of a movie taken, so we can't show the video here, but we can basically analyze individual cell and build and trace and ratiometric trace. And you see with the FRB Untitled FKBP Halotech system, you can trace these individual cells and you can see a beautiful response after addition of rapamycin that is time dependent. On the other side with FRB ALAC 8 FKBP YFP, there's very little and very noisy response to be observed. Next. Now I want to give you a few application examples. We didn't, the FKBP FRB system is of course a somewhat artificial system and not really relevant, doesn't really cover any relevant biology. So we wanted to just touch upon a few actual protein-protein interactions that might be of some physiological interest. The first, we used it for measuring nuclear hormone receptor dimerization. Next. Of course, upon the addition of an agonist, these nuclear hormone receptors dimerize, homodimerize, or some of them heterodimerize, and some of them transfer then in the nucleus. So we tested this with the estrogen receptor, and you see a nice dose response in change of BRET ratio in response to beta-estradiol. Next. And we did it also with the glucocorticoid receptor, and upon addition of a dose response of dexamethasone, you see an increase in BRET ratio. Now the interesting thing here is, next, you can actually combine that with an image-based analysis, and that just shows you the Nanoluck channel. So one of the fusions, the glucocorticoid receptor Nanoluck fusion, and you see the untreated cells where the signal is mostly in the cytosol, but then upon dexamethasone treatment, within about 10 minutes, most of the signal migrates into the nucleus. That gives you just another way of validating your assay in an independent methodology, and that makes Nanoluck so valuable in this context. Next, please. Another way of applying this, applying NanoBREad, is the building of sensors for biological or physiological processes. One would be, for instance, caspase-3 as an indicator, as a lifestyle indicator for activation of caspases. It's simply a Nanoluck Halotech a fusion that is separated by an DVD site that is caspase-3 sensitive. Upon caspase-3 activation, this construct would be cleaved and you would see a lot of BRET. Next. You see here an example where we treated cells with increasing concentrations of staurosporin. You see a nice dose-dependent decline in BRET following staurosporin treatment. You see the same if we treat cells with trail. The differential is not as large, but you still see a nice dose response. Next slide. Now, if we compare that to BRET1, next, you see that with BRET1 you see the same dose-dependent decline. However, of course, the plateau is much higher because in this case, if you show it as uncorrected BRET, you basically have a large background to contend with. Another important aspect here is, again, the physical stability of Nanoluck compared to ALAC 8. And you see, if you compare just the luminescence output of both enzymes, that Nanoluck is not directly affected by the undergoing apoptosis. However, ALAC 8 seems to get degraded as apoptosis goes on, or if you have a stronger apoptotic input. You don't really want your reporter to be influenced by physiological processes, so in that regard, you want a reporter that is stable independent of the physiological input. Next slide, please. So another example I want to quickly present here for protein-protein interaction is the use for looking at the interaction between GPCRs and beta-arrestin 2. And of course, if you treat a GPCRs or number of GPCRs with an agonist, it will recruit beta-arrestin, either beta-arrestin 1 or beta-arrestin 2, to its C-terminal cytoplasmic tail. In this case, we fused the different GPCRs to Halotech and beta-arrestin 2 to Nanoluck. And you see, we show you a couple of examples here. Here's vasopressin receptor fused to Halotech and beta-arrestin to Nanoluck. see a nice dose-dependent increase in BRET following the administration of serial dilution of vasopressin. The same happens if you use, instead of vasopressin receptor, beta-2 adrenergic receptor, and serial dilution of isoprotenolol produces a nice dose response, an increase in BRET. Next slide. Now, once again, we can use actually imaging to corroborate some of the results. So in this case, we can correlate protein translocation and BRET in the vasopressin receptor 2 and beta-arrestin 2 model. So in the first panel, you see untreated cells. You see bright donor but little acceptor signal. In the second panel, you see vasopressin-treated cells. Not only see you now the acceptor coming up, which is the AVPR halotag fusion, but you see also the characteristic pitting, so indicating that actually the physiology of this reporter is intact. And then next, you can basically use individual images to trace individual cells and their response. Once again, this was part of a video taken over time. And you see the vast difference between the reactivity of different cells. And I think this is an emergent theme why imaging or image-based cytometry becomes important, is that even a clonal cell line, if you look at a clonal cell line and you measure the reaction of individual cells against a stimulus or inhibitor, you get quite a bit of divergence between different cell lines, which might depend on a number of physiological factors. But that is important information to capture and can make overall analysis of physiological processes much more precise. Next slide. So for the sake of time, I will take this slide next. One interesting assay we started building, and this is work that was done by Danette Daniels, who is a colleague of mine here at Promega. And they basically became interested in the interaction between histones and Bromo domain proteins, which of course plays an enormously important role in epigenetic regulation. Now, this interaction is static, and we show it here, the interaction between histone H33 and H4 with Nanoluck BRD4. And you see basically a single bar indicating an interaction as an increase of BRET. Now, ideally, we want to have the ability to change that interaction to make really sure that it is actually a physiologically relevant interaction, but also to build an assay that potentially can use the disruption as interaction for therapeutic purposes. Next slide. And so, next. So they treated these samples with a number of known known agents that interrupt the interaction between BRD4 and histone H3. JQ1 out of Bradner's lab would be one. And they're fairly specific for the Bromo domains of BRD4, a number of related proteins, but not other proteins. And you can see that difference clearly here if we look at measuring the interaction between NLUC BRD4 with histone H3 or NLUC CBP with histone H3. While CBP is largely unaffected, NLUC BRD4 gets displaced from histone H3, and the same can be observed for histone H4. Next, please. Now, this gives you a brief overlook about some of the work we have done in the past, showing you how NanoBRET can be applied to a multitude of different applications. And hopefully I convinced you that NanoBRET poses a considerable advantage over the current BRET systems. I want to conclude with a number of general considerations for NanoBRET in cell-based assays. First of all, you always should look for basically the best BRET pair by the means of empirical analysis and not make any assumption. It is ideally that you test multiple donor-acceptor DNA ratios to find the optimal range. Test also multiple DNA or concentration or expression levels to see if you're in a physiological range for your system. Of course, endogenous expression levels need to be taken into account because if endogenous proteins pair with your reporter, that will result in a NanoBRET interaction. As far as instruments go, we generally recommend filter-based instruments because we need to capture as much light as we can, even with the NanoBRET system. Monochromator-based instruments, they can work under extreme conditions where you have very high expression levels and very large samples. However, in most cases, they won't work satisfactorily. Now, the exception to that is the BMG Clarius star, and you basically want to take this opportunity, lead over to the next presentation. I want to just show a couple more slides next. This is the vasopressin HT beta-arrestin 2 model. And you see here the Clarius star using the monochromator settings produces a beautiful dose response to vasopressin. You see nice dose-dependent increase of BRET. If you use a monochromator and a couple of other monochromator-based instruments, you see either what amounts basically to noise or a very noisy dose response. Next. Now, if we compare, we use in this case the FKBP-FRB model, the Clarius star to a filter-based instrument. The Clarius star, you see once again, produces a nice dose-dependent increase of BRET to rapamycin in response to rapamycin. And we use a filter-based instrument, and you see that the quality of data is essentially equivalent. Next slide. I would like to conclude my presentation here by giving credit to a large number of my colleagues here at Promega. The list is too long to read, but it's needless to say that plenty of people are involved to make this work possible. Thank you very much, and I would like to hand it over to EJ.
Okay, thank you very much, Thomas. That was a great introduction to a presentation on the NanoBRET assay. And I also want to thank Tim Stutt for the nice introduction to this webinar. As you've seen already with the NanoBRET assay and some of the data presented by Thomas, the Clarius star is a pretty unique instrument in that its monochromator is able to do some advanced assays like BRET assays, FRET assays, and some of the other assays that classic monochromators cannot do. So what I'm here today to talk about is the new this new multifunctional multi-mode microplate reader from BMG LabTech. It is the Clarius star. It is a high-performance reader which has this new unique technology known as LVF monochromators. A little background in microplate readers before I delve into the Clarius star to set a little precedent. Microplate readers have been around for about 25 years, and in that time there are some evolutions in microplate readers. One of the first evolutions in a microplate reader was going from single to multi-mode instruments. Now one of the most common microplate readers out there is a three-mode reader that can measure absorbance, fluorescence, and luminescence detection. BMG LabTech was one of the first microplate reader companies out there to make a multifunctional reader, and so we have a lot of experience in making multifunctional readers. And in that experience, we learned that when you have a multifunctional reader with different detection modes, it's a challenge to not compromise between those different detection modes. And that's a recurring theme that I'm going to come back with the Clarius star, is how the Clarius star does not compromise between the different detection modes in this multi-mode reader. A second evolution in microplate readers was that classically where they were just used to measure simple biochemical assays, but now they're used to measure more complex heterogeneous cell-based assays, such as Thomas was talking about with the NanoBRET in living cells. And as part of that evolution of microplate readers, new technology had to be introduced to microplate readers. One example of that technology was an atmospheric control unit that can regulate both CO2 and O2 independently within the microplate reader. So again, a nice evolution in microplate readers. Our next evolution in microplate readers was to incur different technologies to help it do more and more complex assays applications. And then a third evolution occurred in microplate readers in the last 5-10 years, is that not only are they multifunctional or multi-mode readers, but they actually have different detection technologies to do those different detection modes. For instance, if you were doing fluorescence density, it can be done with either filters or monochromators. So when you were purchasing or going to use a microplate reader, one of the questions was, do you want the sensitivities that filters offers, or do you want the convenience that a monochromators offers? So that was one of the new next evolution in microplate readers that allowed new technologies. Another example of that is absorbance. In microplate readers now, you can do absorbance 3 different ways, either using filters, monochromators, or a spectrometer. So again, a next evolution is newer technology that was added to microplate readers to help them to do the assays differently. Another example is lasers that were added to help for advanced assays like TRF and alpha technology. So saying that, giving a little history of microplate readers, we believe that the Clarius star is one of those next jumps, next evolutions in microplate reader technology. With the Clarius star, you can do anything as possible. You can capture any wavelength, any bandwidth, and you can perform any assay with the Clarius star microplate reader. Like all good multi-mode readers, like all good high-performance readers, it is a multi-mode reader. It can read all of the standard non-radioactive technologies out there. As I talked about earlier, it actually has 2 different technologies to allow it to read fluorescence and luminescence mode. Those 2 different technologies are a monochromator, this LVF monochromator, filters, or actually a combination of both. This is one of the only microplate readers where you can use a filter for excitation and the monochromator for emission, or vice versa. So again, it allows you to not compromise between those detection modes in fluorescence or luminescence by allowing you to use filters, monochromators, or both at the same time. Being a nice, talking about that LVF monochromator, that why is it so good? You saw some data already from Thomas from Promega about the NanoBRET assay. He said one of the exceptions to the monochromator-based instruments is that the Clarius star can perform the NanoBRET assay fantastically. With that, it has filter-like performance. It has adjustable bandwidths from 8 to 100 nanometers for excitation and emission, and it has a fantastic limit of detection of less than 0.5 picomolar, or 10 attomoles per well. So it's a next-generation monochromator that has filter-like performance in its flexibility and in its sensitivity. And then another third part of the Clarius star's technology, or technology found in this reader, besides its LVF monochromator and its filters, is its spectrometer. So what that does is it rounds off BMG's triple detection technology in the Clarius star. That is filters, spectrometers, and LVF monochromators. We use the spectrometer for UV absorbance measurements, and that allows you to do instantaneous spectral scanning. You can capture 220 to 1,000 nanometers. That's 780 individual wavelengths in less than 1 second per well. Classic microplate readers use filters or monochromators. You only capture 1 wavelength or 1 or 2 wavelengths in the same amount of time that the spectrometer can capture 780 individual wavelengths. So it's a nice technology to do absorbance in the Clarius star, and it rounds out the triple detection technology of filters, monochromator, and spectrometer. Just a couple other points on the reader before I get into more detail about this new technology found in the reader. It has reagent injectors to allow you to do kinetic and cell-based assays. It is a fantastic reader for FP, one of the best readers out there as far as standard deviations of fluorescein. So if you want to do advanced assays like FP, the Clarius star can do them really well. It's also very good for luminescence, as Thomas has pointed out in the NanoBRET assay. But one of the nice features in the Clarius star is actually has a 7-log dynamic range for luminescence detection. A lot of other readers out there will max out at 6 million, 7 million relative luminescence units. The Clarius star can go up to 10 million relative luminescence units. If you compare it to some of those other readers, you have a 75% larger dynamic range on the high end of your luminescence reagents. Luminescence assays. And just as a little summary of some of the specifications for the Clarius star. The one thing I would like to point out at the very bottom is the read times. You know, the sensitivities are very good for the Clarius star, and the read times are also fantastic. It would may not be as fast as the high throughput screening instruments out there, but it definitely can be used for any assay development laboratory or for mid throughput screening if that is needed. Okay, so one of the new and unique things about the Clarius star is its LVF monochromator technology. So what is this LVF monochromator technology? LVF stands for linear variable filters. Linear variable filters are just what they say. They allow light to go. They allow light to pass through the filters above or below a certain wavelength. And what the linear variable filters, you have a short pass and a long pass that can be overlaid on top of each other. And if they're properly aligned, no light can go through. But as those linear variable filters, the long pass and short pass, are moved relative to each other, you can create different wavelengths and you can create different bandwidths. So completely new technology than classic monochromator or grating-based monochromators. So the LVF monochromator is based on linear variable filters, and so it's a completely new type of technology that can be found in monochromator, that can be found in microplate readers. So why are the linear variable filters, the LVF monochromator, so much better? One of the main reasons is that they transit more desired light to the detectors. For excitation and for emission, they allow more desired light to be passed through than grating-based. Why do they do that? Well, one of the main reasons is that there are no stray light. It's completely different technology than grating-based monochromators, so it doesn't have to account for the inherent flaws that are found in that technology. So it doesn't have to count for the stray light, or it doesn't have to compensate for a stray light. And the second nice feature is that it has a linear variable dichroic mirror. This is the only instrument out there that has a linear variable dichroic mirror. A dichroic mirror allows wavelengths at a certain wavelength that allows the light to pass through above that wavelength, or blocks light below that wavelength. For instance, if you had a dichroic mirror of 490, all the light above 490 would be passed. All the light below 490 would be blocked. With a linear variable dichroic mirror, you can actually have that varied over a broad range to increase the sensitivity of the monochromator. And then a nice feature, and probably the most applicable to end users, is that it has broad bandwidth up to 100 nanometers. Doubling bandpass equals 4 times more light, so you can just see by increasing bandwidth from 20 to 40 nanometers, you would get 4 times more light, so therefore increases the sensitivity of these LVF monochromators. So those are the main reasons why this new technology is much more sensitive than grating-based monochromators. Just to give you a representation of the LVF monochromators, there's one for excitation, one for emission, and they're separated by a linear variable dichroic mirror. As we blow up that linear variable dichroic mirror, you can see that the slide can just move along depending on the wavelength that is automatically chosen, and that helps increase the sensitivity of this LVF monochromator dramatically. As I said, one of the main features is that it has continuously adjustable bandwidths from 8 to 100 nanometers. Why is that important? Well, existing monochromators only go up to about 25 nanometers, or they have adjustable from 22 nanometers. So this has 4 times larger adjustable bandwidths than any other monochromator on the market out there. Why is that important? Well, some assays just don't work. As my colleague Thomas just pointed out, the NanoBRET assay does not work well with monochromator-based instruments, and they highly recommend filters. Other assays, such as dyes that emit very broadly, fluorescent protein dyes, also dyes that emit in the far red, and tests with 1 or more dyes are very hard to optimize using a limited bandpass monochromator. So broader bandwidth allow more light and greater sensitivity for all assays that you'll could be measuring with the Clarius star. As an example, here is naphthofluorescein with some wide excitation and emission peaks with a 9 nanometer monochromator. You can see that the assay would work, but you got very little excitation and emission energy coming out of it. If you had a fully adjustable bandwidth up to 100 nanometer, look how much more energy you could get for the excitation and for emission out of this particular assay if you used naphthofluorescein. Another particular example is how many users are getting nice pictures with a fluorescent protein assay on their microscope, and they want to take it to a more of a high throughput or get greater numbers to do statistics. So they want to do greater populations in a cell-based microplate assay. So when they translate from a microscope to a microplate reader, you can't do that all the time. You can't use a lot of the recommended settings that are recommended by the manufacturer, the microscope manufacturer. An example of that would be blue fluorescent protein. This particular microscope manufacturer would recommend exciting with 37 nanometer bandwidth and measuring emission with a 75 nanometer bandwidth. With common monochromators, you couldn't do that. With a Claristar, you can. And then exactly a third example is the CFP, YFP FRET assay. As my colleague said, it's like making your own filters. You don't have to compromise on -10, -20 filters and have to make your assay work with what filters you have or what's the limitation of your monochromator. With a new LVF monochromator, you don't have to compromise your assay based on your technology. All right, so the LVF monochromator is a really nice feature to the microplate reader, but it's not to the Claristar, but it's not all that it has. One of the nice features of the Claristar to help you do assay development and to help you run the instrument is a nice filter selector viewer. We have an integrated fluorophore and lumiphore library in our software. It's just a drop-down, pull-down menu, and all you have to do is click and drag them, the appropriate filters or monochromator settings according to how you want them. And if you needed to add a second dye to your spectrum to see how they would overlay, you can do that in the software. So a very nice feature in the software is an integrated lumiphore and fluorophore library that allows you to optimize your assay very easily using the monochromator with adjustable bandwidth and adjustable wavelengths. As I said earlier, it not just has the monochromator, but it also has filters. We included filters just to ensure that we never compromise in any of the assays that the Claristar can do. Filters will always give the best performance in TRF, TR-FRET, alpha technology, and FT assays, and that's why we've included it in the Claristar. And then a nice feature with the Claristar is the mix and match of the excite with a filter and measure emission with a monochromator. Very nice feature in the microplate reader, in the Claristar microplate reader. Talking about that sensitivity, here's two other readers in BMG's line. The FlowStar Omega has been out in the market for over 4 or 5 years, and then our Ferristar FS. Both the FlowStar and the Ferristar are filter-based instruments, and you can see where the Claristar stands relative to the limits of detection for fluorescein with its monochromator and filters. So you can see that the Claristar filters are just as sensitive as a high throughput screening instrument, and actually you can see that the Claristar's monochromator is more sensitive than other instruments, filter-based instruments. So it's a fantastic detection for fluorescence sensitivity with using both filters and the monochromator. In summary, I'm not going to hit this slide in summary. I'll just put it up there so you can have it. So it's a fantastic multi-mode reader that does not compromise in any of the detection nodes based on its triple detection technology and all of the integrated technology inside of the reader. Sorry about this. So as we say with the Claristar, any wavelength is possible, any bandwidth is possible, and any assay is possible with this next generation monochromator and next generation BMG lab tech. I would like to thank my colleagues in Germany, our team of engineers. They did a fantastic job on this instrument. They always amaze me when they can come up with a fantastic technology that is new and novel to the microplate reader market. I also want to thank our BMG application scientists for all of the nice data that they presented to us, and I also want to thank Thomas and Promega for sharing this webinar with us, and I want to thank Tim Stute and DDD for hosting the webinar. Without further delay, we have 5 minutes left, and we can take some questions.
Thanks, EJ. If you want to go through some of the questions that are shown in the queue there, you can go ahead. Either you or Thomas can respond to them.
Okay. So the first question is, how do you interpret the interaction in cell-based assays to specific effects in terms of potencies? And this is to Thomas for Promega.
Sure. I think there are a number of different ways to go about it in context of the BRET assay. And it really depends, by and large, on the biological system you're using. I think, of course, if you have, for instance, incomplete activation, or you have simply different strengths of agonists, and that probably applies in particular to the GPCR world, you can attain different amplitudes in signaling between the different treatments. Another way to look at the potencies of individual compounds, if they have, let's say, different EC50s, you will, if you're able to draw a complete dose-response curve, you can establish rank order potencies. I'm not sure if that completely answers that question, but that's basically what we found in the past in our model systems.
Okay, thank you, Thomas. A second question is for me. Why would a user want to use a filter and a monochromator in the same measurement? Good question. So as I mentioned, the Claristar has two different detection technologies. It has the filters and it has the monochromators. And it's one of the only instruments, or the only instrument out there that can use a combination of two instruments. Why would you want to do that? Well, some assays, say you're exciting down in the UV range, you may want to use a small, narrow bandwidth filter of 2, 3, 4, 5 nanometers. And then you would want to use a broad bandwidth using the monochromator. Or you have a specific filter and then you want to do assay development with the monochromator, and so you can excite with your specific filter and then you can do assay development with your monochromator. Another nice feature with a mix and match is you can actually do excite with the filter and scan with the monochromator. So again, you can optimize your assay and you don't compromise for your assay never compromises because you have the option to always use filters, or you can use the more flexible monochromator. So that's why it's important to have. That's why we've included both monochromators and filters. Okay. Is there any other questions? There's one more question. It said, can you explain further how the LVF monochromator separates light using the linear variable filters instead of a metal grating? Well, I mentioned it earlier, and we can give you more detail because we only have a minute left before we have to end the webinar. But rather than having metal gratings that separate light out into the different wavelengths, as I said, it has a long pass and a short pass filter. If you can imagine a long pass, short pass filter, if they're properly aligned, they will block all light out. So if I had a long pass filter for 490, it would let all light in for 490. If I had a short pass filter for 490, it would let all light in for under 490. If you properly align those at 490, then it blocks all light above and below 490. But if you slowly separate them out from each other, you can actually increase 490, 492, 493, and get a nice little bandwidth around a certain wavelength. So that's how the linear variable filters work differently than the gradient-based monochromator.
Alrighty, and it looks like we're pretty much out of time here. I'd like to thank EJ. EJ, if you could forward the slides to that one survey slide, should be next to the last slide. I'd appreciate it. And we'd like to I'd like to ask the audience to respond to the survey there on your screen, and like to thank Thomas and EJ for their participation today. And if you have any questions or anything, you can forward them to either Drug Discovery Magazine or to EJ or to Thomas at BMG LabTech. And we'll get those questions responded to. Again, thank you very much for your participation today. And this This recording will be live on our site within 24 hours, and for online viewing for the next for more than 6 months, anytime during the day, 24/7. So thank you very much.